Mitosis is a kind of division in which one cell, called the parent cell, divides into two new cells that are genetically identical to each other and to the parent cell. Mitosis is used in most cells in the body, which are called somatic cells, for growth and injury repair. Before the cell can go through mitosis, it grows in size, makes sure it has enough nutrients and resources for division, doubles its DNA, and replicates its centrosomes, two organelles made up of two centrioles each, in a phase called interphase. The first step of mitosis is called prophase. In this step, as the nuclear membrane begins to disappear, the cell condenses its loose DNA or chromatin into chromosomes, structures of tightly coiled DNA and protein. Each chromosome is made up of two sister chromatids that are attached at the centromere. Also, the mitotic spindle microtubules begin to form, extending from the centrosomes, and continue to grow as the centrosomes move away and towards opposite poles of the cell. Next step is metaphase. As the centrosomes near opposite poles of the cell, the mitotic spindles attach to the centromeres of the chromosomes as structures called kinetochores. As the centrosomes reach opposite poles of the cell, the chromosomes are pulled towards an imaginary line at the center of the cell called the metaphase plate.
In anaphase, the microtubules attached to the kinetochore shorten, pulling apart the sister chromatids in each chromosome. The centromere of each chromosome breaks down to allow for this division. Other microtubules that attach to the centrosomes in each other lengthen, pushing the poles of the cell farther apart. During telophase, the sister chromatids, now called chromosomes, reach opposite poles of the cell, and the division of genetic material is complete. Each half of the cell contains one complete set of DNA identical to that of the parent cell. Then the mitotic spindle breaks down and new nuclear membranes form around each set of chromosomes, forming two new nuclei. Once the membranes are formed, the chromosomes begin to unravel back into chromatin. In the final step of mitosis, called cytokinesis, a structure called an actin ring forms in the cell, which constricts the cytoplasm inward, forming an indentation called a cleavage furrow. Then the cell divides its contents equally between each side of the cell. The actin ring continues to constrict the cytoplasm until the cytoplasm finally divides.
Two daughter cells are completely identical in size, cell contents, and genetic material, both to each other and to the parent cell.